that Darwin is famous for asking about in his book On the Origin of Species, which is, how do you define species and how do species get created? So here we have two different species, the horse and the donkey. We know, as biologists, that the horse and the donkey are two different species because when we mate them together, they make the mule. And mules are sterile. And so that's one of the ways that we know that these are two different species. If they can't have offspring together, or if they are mated together and they produce offspring that are sterile, these are two different species, horses and donkeys. In this case, we know the genetic reason why they're two different species is that horses and donkeys happen to have different numbers of chromosomes. So the DNA content in these two species is different, and that poses some real problems for the mule. And a lot of people might think about the mule as a useful farm animal, but it also has this terrible problem, which is that it's sterile. I actually look at it as sterile being a good trait. And so one of the implications or applications that I'm going to try to convince you of in the next 3.5 minutes is that it's interesting to understand the genetic basis, the genes that cause hybrid sterility, because we might be able to use that for biocontrol. In my lab, we study mitochondria, which are components of our cells that produce the energy that cells need to survive, essentially. And there are particular proteins, which I'm diagramming here as sort of gears. One of these proteins, or several of these proteins actually, are produced by the mitochondrial DNA itself, which I'm diagramming as circles. The other components, proteins, are encoded by DNA that are found elsewhere in the cell, in the nucleus. And for the rest of us, we can basically think of this protein machinery, this complex, as converting the food we eat into cellular energy that our cells use. And all of this has been funded so far, thankfully, by the NIH as well as a couple other more local funding sources. And what we're trying to do is understand how taking the mitochondrial DNA, or one of those gears, from certain populations of organisms, like these microorganisms or microscopic worms, C. briggsii, that I work with. We have populations that are found in Japan and in India. They're going to be color-coded now, red and blue, for the rest of the talk. What happens if we take some of those cellular components, the proteins that do energy production, from the blue population and combine them with other proteins that come from the red or the Indian population? Might that have an impact here in the middle on what happens during energy production? So in the Indian strain of C. briggsii, they're happy, they're healthy, they're fertile, they produce offspring. The same is true for the Japanese population, but what happens when we make this artificial hybrid where we've taken these chromosomes from the Indian population and this chromosome from the Japanese population, combine them together, and we get this weird blue and red combination of proteins, cogs, gears. Does that actually function? And we've got a lot of data, but you don't need to see it, and you probably don't want to see it. So here's just one data slide showing those different combinations of genes. So we've got the Indian parent, and what we're measuring here is the number of offspring that they produce. This measure of fitness, how healthy those worms are. And if we look at the Indian, the native, the wild population from India and from Japan, they on average will have about 200 offspring or so, and that's fine. But when we make this mispairing artificially in the laboratory by hybridizing these two populations together, the number of offspring they have drop. They're not totally sterile, that is, these are not the mules of the worm world, but they're getting close. And so what we can do, because they're not totally sterile, is we can actually go in and start to use genetics to understand what are the genes that are on those red and blue chromosomes that do cause these hybrids not to be able to reproduce as well as their parents. So in other words, horse and donkey are happy, their offspring is unhappy, and we've found basically the same thing using an animal we can bring into the Jordan building. C. briggsii, like their other relatives, C. elegans, are not pathogenic to any organism that's known, and they're certainly not crop pests. So as I've mentioned, one of the main aspects of this could be biocontrol. Another one might be the genetics of species identification. So I'm a geneticist in the biology department, and I'm very happy to pursue any sort of collaborations, especially if you think you need some assistance with genetic identification or genetic testing. And I'm glad that I no longer stand between you and lunch.
Actually, I'm going to suggest that Dr. Willett would go through yours.
Oh, sure.
Right?
Of course. It's an advantage that we are.
Good, now you stand between us.
But the thing is that you already introduced some stuff for me, so.
Oh, it's perfect. Happy to help.
Thank you.